we have 4 speakers from RUMIGEN and GEroNIMO, and we have an invited speaker, Karel de Greef from Wageningen University, and he will put a different focus on our discussion. So now, I don't know if Guillaume is in. Otherwise, I'm going. Okay, okay. Now I give the floor to Guillaume then.
Thank you, Shara. So you should be able to see my screen now.
Yes.
So hello everyone, I'm Guillaume Devailly, and I'm really happy to present you this prospective review we did in the GEroNIMO project. So in mammals and most vertebrate species, gene expression is under multi-layered control, and notably involving epigenetic mechanism and epigenetic marks. And as epigenetic marks, we means reversible modification that happens on the chromatin fiber, either directly on the DNA, such as DNA methylation, or in histone tails, such as histone modification. And maybe epigenetic marks could include also chromatin accessibility. So epigenetic marks, they can be deposited on the chromatin by epigenetic writers. They can be removed from the chromatin by epigenetic erasers, and they need to be interpreted by proteins that we call epigenetic readers. And this is the case, as I said, for DNA methylation, DNA hydroxymethylation, histone post-translational modifications, and to some extent for histone variants. Classical approach to study the link between epigenetic marks and gene expression involved the comparative mapping of epigenetic marks across different animals and biological conditions. And this is what we do a lot, for example, in GEroNIMO, but it leads only to correlations between gene expression level and gene expression, and we don't know if it's the changes in epigenetic marks that cause changes in gene expression, or if it is the changes in gene expression that cause changes in epigenetic marks. And we have good molecular evidence that both mechanisms could happen. So people have been doing a lot of knockout, knockdown, overexpression of epigenetic writer, eraser, and readers to try to better understand their function. And also chemical inhibition of epigenetic enzyme have been used, but both of those approaches, they affect the whole genome, so it's not easy to discriminate between primary effects on site and secondary effects that are due to whole genome alterations. That's why people have also been developing epigenome editing approaches. By epigenome editing, we mean the targeted modification of one or several epigenetic marks at one or several genomic locus. And epigenome editing can be used to investigate the causal mechanism linking epigenetic marks and gene expression regulation, or to investigate, to propose some experimental validation of distal regulatory elements that have been identified otherwise. But also, epigenome editing could be used to modify a target gene expression level without modifying its DNA sequence. For example, here, if we have the case of an active promoter that is unmethylated both on DNA and on lysine 9 of histone 3, and this active promoter leads to the transcription of some transcripts, we could deposit some DNA methylation and histone marks using epigenome editing, and those newly deposited epigenetic marks could induce transcription repression on the target gene. Also, the tools between epigenome and genome editing are mostly the same, and the name also looks a lot like the same. I would like to highlight quite a few important differences between epigenome editing and genome editing. With epigenome editing, we use, for example, inactivated Cas9 that no longer cut the DNA. And so there is no cleavage and no mutation induced in the DNA sequence with epigenome editing. Because of this, we also target a slightly broader region. With genome editing, we can change a single base. With epigenome editing, we target a broader region. For example, for DNA methylation, we expect a few hundred base pairs to become hypermethylated. And it's the same. We are expecting to target a few consecutive histones for histone tail modifications. A consequence of that is that epigenome editing is probably not stable in most contexts, with spontaneous reversions that could occur in days or weeks. However, in some contexts, stable epigenome changes have been observed, either in vitro or in adult somatic tissues. Some people have also been doing genomic insertion of epigenetic editing tools, which to trigger more long-term effects. And lastly, I wanted to highlight that even transient changes of gene expression can still be useful and can still have long-lasting effects on the life of the animals. In GEroNIMO, we did a review of existing studies in vivo epigenome editing, and what we found was mostly studies in mice, with one study in chicken embryos before hatching. And these studies, they used a variety of epigenome editing tools and a variety of delivery methods. And some of these studies, the methods they chose resulted in alteration in the DNA sequence of the cells, but in some others, there was no alteration of the DNA sequence. So to do epigenome editing, you need a guidance system, you need some epigenetic effectors, and you need some delivery methods. For the guidance systems, the historical approach consists of the combination of various zinc finger domains or various tails domain. And it's the combination of all those domains that leads to sequence specificity. So each time you want to change a target, you need to reassemble all those zinc finger domains, those DNA binding domains, to create a new sequence specificity. Since the development of the various CRISPR-Cas9 technologies, people have been using modified Cas9 for the targeting on DNA. With a canonical Cas9, there is a guide RNA that targets the target sequence by sequence complementarity, and the Cas9 will then cut the DNA at this loci. In epigenome editing, we use inactivated or dead Cas9, so the guide RNA still targets the Cas at the locus by sequence complementarity, but the Cas no longer cuts the DNA. Maybe one drawback of the Cas9, the canonical Cas9, is that it's relatively long, 4 kb long, so this could have an impact on various delivery methods. So people have been developing various alternative Cas by making them shorter, but those alternative Cas might have a different structure of the guide RNA, and so the commercial availability of guide RNA for those alternative Cas might be more limited. A variety of epigenetic effectors are merged to those guidance systems. So people have been using, for example, for DNA hypermethylation, they've been using DNA methyltransferase, such as DNMT3A, its helper DNMT3L, or a bacterial DNA methyltransferase named MQ1. Those methyltransferases can be coupled with other repressive domains from CRAB or MECP2 as popular choices. For gene activation, people have been using, for example, for DNA methylation, the TET1 enzyme, and as activation domain, the VP64 or P65 or the RTA domain. Those activators or repressors are merged with the targeting system, for example, as a form of a fused dead Cas9 plus DNMT3A. Or sometimes it's a modular design where the dead Cas9 has a tail of molecular tags, and then the epigenetic effectors are coupled with anti-tags, and the complex is assembled within the cells. Several delivery methods are used in the literature, either DNA lipoaffection, RNA lipoaffection, ribonucleoprotein lipoaffections. Those approaches are quite simple, but can suffer from limited transfection efficiency in some context. Also, for RNP liposuction, you need to have your purified proteins in vitro, and the availability of purified proteins for epigenome editing is more limited. You need to produce them yourselves because they are not non-commercially available for now. Some people have been using electroporation or nucleosuction, which is mostly in vitro. In vivo, it can be used for the chicken neural crest on the chicken embryo. And in mouse, you can do electroporation of embryonic stem cells and then inject them back to mouse embryos to produce chimeric embryos. Other developed tissue might be accessible in vivo, but this will require some surgery. In mouse, some teams are doing genome integration of the epigenome editing tools, so they are expressed in all the cells of the organism, but this creates genetically modified organisms. Another approach is the use of viral delivery through lentivirus, adenovirus, or AAV. And those viruses, you need some sanitary measure to manipulate them. And some of them, it might lead to host genome modification with the integration of the content of the virus. I would like to highlight maybe one sample example of a study of in vivo epigenome editing in mice. A team from Italia is using this epigenetic editing tool with a dead Cas9 fused with DNA methyltransferase and transcriptional repressor domain. This complex is guided at the PCSK9 promoter using a guide RNA. And the team has compared epigenetic editing tools, so this construct, CRISPR inhibition, which is just the dead Cas9 plus the crab domain, and canonical gene editor, so wild-type Cas9. Those tools have been encapsulated in a lipid nanoparticle, and what they use is messenger RNA coding for those proteins, plus the guide RNA within the LNP. A single injection in the vein of the tail of some mouse, and then the team has measured the PCSK9 protein level in the blood. And you can see for both epigenome editing in blue and genome editing in yellow, there is a diminution of PCSK9 level in the blood, and this diminution lasts at least 1 year. This is compared to the untreated mice in gray. And for just CRISPR inhibition, at first there is a diminution of PCSK9 in the blood for a few days, but then it goes back to normal after a few days. Those LNP, when they are injected in the blood circulation, they mostly act in the liver. So the team has also measured the PCSK9 messenger RNA level in the liver of those mice. And indeed, both epigenome editing in blue and genome editing in yellow can induce stable repression of the PCSK9 messenger RNA in the liver. So as far as I know, there is not yet a lot of in vivo application for epigenome editing tools in farm animal species. Nonetheless, the tools, the molecular tools, targeting systems, effector, and delivery are getting better and better every year. The main application at the moment is mostly for academic use for functional genomic studies. If we see some application in farm animals, they will be highly dependent on the target genes and the delivery methods. Some might want only to target somatic tissues, but some teams would want to target germinal cells to have maybe transgenerational effects. In vivo commercial applications in farm animal production will be highly dependent on the economic feasibility, the public acceptance, and some intellectual property concern around Cas9 proteins. The ethical aspects of epigenome editing in farm animals are not easily dissociable from the current ethical concern in highly specialized farm animal production systems, so I think each potential application will require its own ethical assessment. Nonetheless, maybe COVID-19 messenger RNA vaccination could be an interesting point of comparison. Indeed, those vaccination, it's the injection of a foreign messenger RNA with medium to long-term consequences. And I think some application of epigenome editing could look like that, with injection of a foreign messenger RNA with medium to long-term consequences. And this demonstrated that part of our society is ready to accept biotechnological innovation if the benefit is sufficiently obvious. And finally, it can provide a first point of comparison for the cost of a treatment, and it will be around 15 euros per dose of treatment. In terms of putative field of application, I think maybe we could see some application in the veterinary field. For example, for pathogen treatment, including preventive treatment of pathogens, and maybe in some pain management situation where pain is non-avoidable. In terms of production system application, maybe an application could be developed in hunger management of stock. And indeed, at the reproductive layer, animals are often put on diet to improve their breeding performance, and maybe epigenome editing tools could help with that. Or as an alternative treatment to hormonal treatments, or for help with egg or sperm sexing in some situations. So as a conclusion, epigenome editing tool, it's the target deposition of epigenetic marks. It's a fantastic tool to better understand gene expression regulation, and it could be used to alter a target gene expression level in vitro and in vivo. It's a fast evolving field and tools. And I think the first commercial application we will see in livestock will probably look more like RNA vaccine or drugs than like genetically modified organism or genome-edited animals. And with that, I want to thank Anne-Sophie, Fabio, Carlos, and Frank for their participation in this work. And thank you very much for your attention. All right, I will stop sharing my screen.
I'm speaking, but I was muted. So thank you so much. Also highlighting the potential applications and maybe a little bit of a hint on how it would start being used in the livestock production. Okay, so if you have questions, please write it to the chat box. We will come back to that. Now I would like to give the floor to Eric. He will present the practical examples from RUMIGEN, including gene editing in goats and sheep, and how these models advance livestock production and animal breeding. Eric?
Yes.
Floor is yours.
So thank you, Chala, and good afternoon to all. It's my pleasure to present to you some example of genome-edited animals, small ruminants that we have produced in the RUMIGEN project. So before starting, I just want to remember you what is the RUMIGEN project. So the main objective of this project is to improve the quantitative genetic tools in different bovine breeds, mainly Holstein, but not only Holstein, and through the addition of new traits. And these new traits are mainly genetic diversity, also adaptation to new environment, and mainly adaptation to heat, stress, and climate change. And in this project, we also try to include to see if epigenetic data could improve these quantitative genetic tools for the genomic selection. And we have also tried to see the potential use of new breeding technique like genome editing. just to say to you that the genome editing is not the main part of the project. There is 9 work package in the project, and genome editing only represent one work package, the VP5, with 4 tasks. The first one was a position paper that has been that could be seen on our website. The second was the impact of the genome editing on the level of de novo mutation, and this task is currently in progress. It's not finished, and we have 2 tasks for some example, practical example of gene editing in small ruminant, not in bovine, according to the length of the project. So I will first present the first example, that is the edition of the PRNP gene in alpine goats. And it's an example of genome editing versus natural crosses versus introgression for a trait linked to animal health. Then the second example will be the slick mutation in sheep, and it represents an example of interspecies transfer of a phenotypical trait linked to heat resistance, and then I will conclude. So for the first example on goats, we have chosen this example because there are very few available example to compare natural crosses and genome editing. And so this is one of these few examples, because in Norwegian goats there is a natural knockout, natural mutations that induce a knockout of the PRNP gene that encodes the prion protein. And so the aim was to introduce this mutation in alpine goat by natural process or by GEO. And then the final scientific aim is to compare some associated phenotypes that is linked to the absence of prion protein, mainly on the immune system and on demyelinization. And so the aim was to compare this phenotype on 3 genetic background, different genetic background: the Norwegian pure one that has been already studied, the alpine one, that animal obtained by genome editing, and a mixed background, so the animal obtained by introgression. And so what is this mutation? This mutation in Norwegian goat has been firstly described in 2012 in this paper in veterinary research by our Norwegian colleague. And it's a single base change, so a T replaces a G, but this T will be a TGA, so a stop codon. So the protein will stop at a very early time, eight amino acids after the signal peptide. So in this goat, there is no more prion protein. So to test the integration, we have import some semen of a male homozygous goat for this mutation, and we have used this semen by artificial insemination to inseminate some alpine goats that are represented in yellow, and so we get some F1 animals carrying heterozygous, carrying the Norwegian mutation, but with a mixed alpine and Norwegian background. So if you want to make a full integration, you have then to cross these F1 animals with a pure alpine animals for many generations in order to, at the end, after many generations, you will get the mutation and a piece of the Norwegian genome that are linked to the mutation. So these animals are declared non-genetically modified, even if they are not genetically clean, because they bring a new haplotype around the mutations that you have introgressed. So Comparing with introgression by genome editing, you use the CRISPR-Cas9 system that I will detail just after. And so you will get some animals that are called founder animals. We should have the Norwegian mutation, but this animal could be mosaic, or there could be also some off-targeting problem. So you have to cross these animals with a wild-type animal to get heterozygous animal carrying only one mutation. And then if you want homozygous, you have to cross this F1 animal to get homozygous animal. So this animal are declared GMO, but you have only create a new genetic variant. So to do this experiment, we have done 8 experiments, and each experiment consists of 5 donor goats and 6 recipient goats. So first, the donor goat and all this animal was cool animal to respect the 3R rule. So the donors are treated for superovulation, and then you recover the zygote, the one cell stage embryo, but it could be also oocyte, that have not been fecunded. So we inject the CRISPR-Cas9 protein with a single guide RNA, and we inject also in this embryo a single strand DNA that is homozygous to the PRP target region in order to bring the Norwegian mutation. So then we keep this zygote for 18 hours in culture in order to see which was fecunded or not. So in order to have a 2-cell stage embryo, and this embryo was replaced on synchronized recipient goat. Then you have to wait for 5 months, and you could have founder animals. And as I said, you have to cross this founder with wild-type animals. So by this way, we obtain 10 newborns in 22 and 23, in 2022 and 2023. Among these 12 newborns, you can see that almost all of them has been genetically modified, except 2 wild-type. But we did not obtain any founder by homologous recombination. So that means that we haven't been able to reproduce the Norwegian mutation. But we get 4 founder with some knockout allele, and among them there is one male that was very interesting because he have only one base and an insertion of one base near the Norwegian mutation, and so this one base insertion normally give rise to a friendship and a knockout allele. So this bug has been used to derive a goat line from him. So if you want more details, this work has been published in Veterinary Research in January this month. And so actually we have produced F1 animal, and then we have produced F2 animals, and actually we have for the knockout allele, we have 4 males and 1 female that are homozygous for the mutation that came from Caesar, from the bug Caesar. We have also a new allele that is a small deletion of 2 amino acids, and that we want to test for the potential resistance to prion disease. So we have 3 male and 3 female. We have some control, and for introgression for the PRPNT, for the PRPNP tier mutation, we have 1 male and 1 female that we will test. So now, in the year to come, we will test the different phenotype. We will first test the resistance to prion by a PMCA test, that is a test in vitro. We will also investigate the gut wall, because apparently there is some differences in the gut wall of prion of goat without prions compared to wild type, and we will test the demyelinization phenotype and also the immune system. So the second example is the transmission of a trait from bovine to sheep. And this example on the SLIC mutation has been done by Simon Lilico in the Roslin Institute. And so I will present you what is this SLIC mutation. This SLIC mutation is a mutation of the prolactin receptor gene, and it's a dominant mutation, and all the this mutation has been observed naturally in different bovine breeds, at least 3 bovines that live in tropical regions, so under hot temperature. And this mutation consists of a truncation. It's not a knockout. It consists of a truncation of the prolactin receptor before, in the C-terminal part, and before this amino acid, that is a lysine, that is required for dimerization and to activate a downstream pathway that include JAK-STAT pathway. And so when the animal, once again, it's a dominant mutation, when the heterozygous animal carried one allele mutate with this variant, so they have a sleek phenotype. This phenotype results in a short hair coat, and this phenotype increases their ability to regulate their body temperature. So the idea was to transfer this mutation from bovine to sheep. And so Simon have get he get homologous recombination, and so he get many sheep with this mutation. And as expected, you can see on the right the sheep heterozygous for the mutation compared to wild type 1, and you see that the sheep have less hair than the wild type during the summer, because it's not true during the winter, but during the summers, you see that there is a big difference. Here I've checked for the fleece weight, for the fleece weight, and you can see that there is a huge decrease of the fleece weight in the sleek animal compared to the control. And we have also done thermal challenges of this sleek sheep. So we have tested 5 animals per genotype, so 5 control and 5 animal heterozygous for the sleep mutation. And you see that the rectal temperature during a thermal challenge, the thermal temperature is lower in the animal carrying the sleek mutation than the wild type. So now in the years that will come, Simon tried to produce homozygous animal for this mutation, and will test them for thermotolerance. So in conclusion, I personally think that in vivo genome editing represents an essential and revolutionary tool in a number of fields, but mainly for basic research, and in order to increase our knowledge in the relationship between genotype to phenotype. But of course, it could be used in agronomy and also in biomedicine. And I want to say that European citizens want an improvement of animal health and welfare, but they do not accept actually genome editing that could represent a tool for this improvement. And if you want a nice example, you can see the example of the resistant to the resistant pig to the porcine reproductive and respiratory syndrome, the PRRS, that some pig has been produced in USA, and they have had the authorization of commercialization by the FDA, and they should arrive soon on the market. So it's a nice examples of how to use genome editing in farm animal. But actually, the problem in Europe is that the European Court of Justice have classified this organism as a GMO. And I think that this decision has put a big stop to potential agronomical project, and by the same way to all the projects, even the fundamental one. And if you want to see our position on this, you can see the RUMIGEN policy paper that is on our website. And so in the future, I hope that the legislation on this animal will be relaxed and that there will be more support around these topics. Before ending, I just want to thank the people who have greatly participated in the goat production, and mainly the private society Alliance with Aurélie Bonnet and Marie-Christine Deloche. I want to thank also all the people who have worked for embryo transfer, the experimental facility from INRAE. We have used 3 experimental facility, and the people involved in the VP5 of RUMIGEN, so Jean-Luc, Simon, Michael, Carol, and our external ethical expert, that is Andy Greenfield. So I thank you all, and it will be my pleasure to answer your question during the discussion.
Thank you, Eric. Thank you so much. It was quite detailed and a broad presentation. Now I have to move on to Matías very quickly because we are really over our time.
You can stop because I cannot stop to share.
Maybe Epa can
Yeah.
help. Okay, so
Okay.
Matías, I would like to give you the floor. We are a little bit behind, so I would just kindly ask you to make it a little bit faster so
Okay.
we
Yes,
don't
that's
give the
fine.
floor to Karel very late. Thank you so much.
Okay. Yeah. Hi everyone. I'm Matías Schrauf. I'm working in RUMIGEN from within Wageningen University of Research, and I will be talking about how we can integrate these gene-edited variants in the genomic prediction models we use for selection in modern breeding programs. So first, there will be, I will talk about why we want that, why we want to integrate this, and what are the problems with that, and then I'll just briefly cover 2 possible solutions, and give some of my conclusions, so that then we can all discuss if you are interested in the discussion later, right? So I will use these abbreviations in the rest of the talk: GE for gene edit variant, and GP for the genomic prediction models, which we use to select animals in breeding programs. So what is the problem? Well, we in breeding programs, we are selecting for many traits, more than we have ever before. So some time ago, breeding programs only selected on some production traits and maybe some conformation traits. But nowadays, most breeding programs, we select for a whole set of concerns. And yet the gene-edited variants are mostly singularly focused, right? They are trying to address the problem, and they might be a very good tool for that, but we still have all the other traits that we are selecting. So, okay, if we decide that gene-edited modification is a way to address one of these concerns, we still don't want to move backward with the other traits, right? So one thing is, well, we can learn from the experience with GMOs, especially in plants, that initially sometimes they had some issues because they modified plants. Maybe they were plants they had in lab, but they were not very good genotypes agronomically. So for example, for yield or some other properties. And then when they had to include those transformed material into the breeding program, that lowered their average yield, and they have problems. Yeah. So we can transform genotypes of animals that we already want in our breeding program. Still, if we modify a few animals, and we want to include them in the nucleus, and we want to increase the allele frequency of the edit allele from those initial founders to the whole nucleus in very few generations, if that sweep is very fast, it can produce bottlenecks. and reduce diversity, and then also compromise future improvement on the traits that we are breeding or traits that we might want to breed in the future. So I hope I convince you that we need to simultaneously, well, if we introduce a genetic variant and we want to increase it in frequency through probably not just one generation, but a few generations, we still want to select for the other traits. And this has some challenges. So if we have this genetic variant that we introduce, and we expect it to be a very causal loss, right? We have a lot of genes and a lot of genetic variation. A lot of it doesn't do anything or doesn't affect most of the traits. But we expect, because we see these big modifications, like from what Eric shown, it should have effects on many traits, right? The cows might eat, or sheep, or whatever. They might eat more, they might eat less. It might affect other traits that we are measuring. And the genomic prediction models that we use are mostly polygenic additive models. So they assume they work well when we have lots of loci, each one with small effect, and also when the changes in frequency of the alleles in one generation to the next are relatively small. And if you stop to think about it, gene-edit variants actually like satisfy none of these three crucial criteria. It's mostly one locus or maybe a few. It has potentially quite a big effect, and we want to increase it in frequency from very little to maybe fixate it in the population. So, plus an extra complication, we have very little information when we start. We might know what it does for what we want to use it, but we don't have good measure of how much it affects all the other trees that we're measuring. So, two potential solutions I will cover, and I will really go over the details now. But one is to modify the priors that we have in our genomic prediction models for the allele effects, especially those of markers that are linked to the gene-edited variant. And this was something that was explored in RUMIGEN by me, together with Yvonne Wenches and Jeremy Vandenplas from Wageningen. Yeah, well, in many genomic prediction models, we have some assumptions for the effects of alleles, which relate to what I said before, that they are small, centered around zero, and more or less homogeneous. And specifically for the ones that are linked to the genetic variant, we could increase the variance. We could increase the mean if we think, okay, they systematically increase, for example, reduce feed intake or increase feed intake or whatever. We could modify their size, their average direction. We can modify both. We have ways to do that in the models quite efficiently. And the first thing we did, well, we explored a very optimistic scenario where we, in a simulation where at some point in generations, we introduce a genetic variant and we see what happens if we select on a genomic prediction model, which is the conventional one, which is this dark purple one. And we look at what happened to the frequency of the edit allele. And we saw, well, what will happen if we modify those models to try to integrate it better? And we saw that, well, if we tell the model that this gene edit has a positive effect on the trait that we're selecting on, then it increases the selection pressure and we select for it, which is expected. But that was the very first scenario. We tried to see if it worked. If it worked, it makes sense to keep on exploring. If it didn't work here, it would not work for anything else. But we have many more scenarios. So these are the two that I just showed. I'll explain in a moment what's this. So here I have in each row one underlying truth, one underlying scenario. So I have, which is what is the actual causal effect of the gene edited variant. And in each column, I have, well, what model we are using. So 0 means the conventional model, and 0.5 means one that assumes that it has a very strong positive effect, right? And we simulated, so each row is a different model, each column is a different model, each row is a different underlying truth. And before we had simulated, like I said, just an underlying truth of a gene edit that favored the trait that was under selection. and we compared the conventional model, which was not changed, that's column 0 here, with one where we assume that it had the effect that it actually had. And yeah, we saw that in that case, we could just increase the frequency of the gene edit allele by using the genomic prediction model. But that was too optimistic. And one problem we saw once we started exploring all combinations is that, for example, if the trait had, let's say the gene edit allele has a positive effect for whatever we are using it, but has slightly negative impact on fertility or on feed efficiency, whatever. What we saw is that here we are putting this prior. We tell the model to first guess that it has a positive effect. And what we see is that the model actually never learns the correct slightly negative effect. It stays with the positive effect. So basically the prior is overwhelming the model. And if we use a slightly smaller, less strong prior, weaker prior, then you see that actually it starts increasing the frequency, but for most simulations that we did, it actually stopped increasing the frequency. That's good. So if we use a weaker prior, then it can adapt to some extent, but not as much as we wanted. So let's jump to the let's jump to the conclusions for this. Is that okay? The prior we see that can help when the guess is not too far off from the actual reality, but the model does not self-correct as fast as we would like. So it could be very beneficial for the initial generations, but behavior in later generations when we have actually more data should be improved. You could just get rid of this prior and go back to the conventional model, and you could do many things in between. For example, there's a thing called fractional posteriors that can be used. With these caveats, this can be used to integrate gene edit to genomic prediction, but it should not be used to select on the gene edit allele to drive the frequency up. So you should have something else in place. Decision procedure, there are ways, we are also exploring this in other projects, where you can drive the frequency of a specific allele in one way or another. But this is just to integrate the effect that the gene edit allele could have on the traits that we are measuring for other goals. There's another possible solution. I'll just say, it's just this is idea that was developed for major genes in Legar and Vitesica 2015. It's to model the major gene, in this case the gene edit, as a trait. So basically, as you have the traits, for example, the weight or the feed intake of the animals, you also have the allele count in the gene locus, for example. And those animals that don't have it have a 0. The animals that have 1 copy have 1, and the animals that have a 2 copy, they're homozygous, they have 2. And that's a new trait. And putting that trait in a multi-trait model helps correct for serious biases and improves prediction accuracy that you would otherwise have if you ignore it. But this also has challenges. So like I said, the GE is initially very rare. Your variance components are hard to estimate. And for a gene edit, you probably also want to consider dominance effects, but that can be added. So all these things could be solved, and this could be quite beneficial in later stages once you have this segregating your nucleus, but you have enough animals with it. So I'm going to the conclusions. We need to manage the frequency of the gene edit allele with other tools than just selection based on the genomic prediction model. So we need to if we go through the effort of doing gene edits, we also need to make sure that they increase in frequency if we want them. So we probably were too optimistic in the initial goals of solution 1, but both of the solutions can still help integrating the gene edit in the genomic prediction model. And this allows us to measure the impact that gene edits have on the traits that we measure. This is useful to keep improving the traits under selection, and also to avoid or monitor unintended consequences that the gene edit could have on other aspects of the animals. And that's all I had to say. I'm happy to go to the discussions later. I'll stop sharing. I hope it was okay.
Thank you very much, Matías, and also making it faster. So now we can move to Karel for the societal acceptance and the responsible innovation part of all these technologies and the applications.
Okay, thank you. Okay, this is not about gene editing, but breeding technology and outcomes more in general. And I will start with a starter, like there's a gap between society and breeding. That's how we started the project, and I will explore that gap and see how we can deal with it. And in two of the previous talks, we already were talking about acceptance a little bit. And my point is that, wait a minute. The choice we made was to explore outside in. If I talk to breeders about our societal role, I will get breeders' answers, but we wanted societal answers. So we interacted quite a bit with people from outside our own networks. This presentation draws heavily on the Breed for Food project, Ethics in Society for breeding companies. And the aim of that project was to understand societal developments. And we did lots of things, and I will acknowledge the team of CRV topics, Hendricks and Wageningen themselves, the colleagues within Wageningen, of course, animal breeding and genomics, for this work. And the highlight of the project we did was to identify who is affecting the societal view on breeding, who do we know of them, and do we know what the role of society is? Do we understand the societal developments as stated in the line above? And then the plan was to discuss with societal partners and to involve them in breeding objectives. And we are at this moment, we are not in the discussion phase, we are in the dialogue phase. So the listening to each other. I will not report on that too much at that moment. I will give some very headlines. And the context is, if you look at the Breeders' Talk Green and other FHAP initiatives, we are always communicating that animal breeding is a responsible task and we take our responsibilities. And at the same time, animal production is criticized quite a bit, especially in Northwest Europe. And the focus is especially on animal welfare, but there's more on animal breeding, which is criticized, or on animal production. And breeding is part of that, of course. And of course, as breeders, we see that husbandry systems change, and we ask ourselves, are we still having the best animals for those new systems? And an example for that is, of course, the fact that pigs are going to free farrowing systems in part of the Western world. And then a breeding company asks itself the question, should I be testing in free farrowing systems to take that as an explicit or an implicit selection environment? And at the same time, we're also, there's a climate crisis in the world, we think, we are convinced of. And there is some adaptive anticipatory behavior in breeding programs on all kinds of sustainability aspects, and the climate thing is one. The example is that a breeding company started to select more breeding companies for methane breeding. So choose those animals for the next generation which are quite efficient in their methane production, low in this respect. And it's not always acknowledged in the outside world that we are doing this. But this is the inside out. This is how we look at it. But we also ask, can you look at it from another perspective to those points? So I'll compare the internal profile we have, how we see it ourselves, and the external profile. And what I was just saying is breeding is doing good. And these are three key arguments. We're successful for food security and resource efficiency as breeders. We're competitive worldwide. We are farmer serving, give them an income. And we do balanced breeding. So we're doing good. If you ask this same question, are they doing good to the outside world, to NGOs? We asked these, animal and environmental NGOs. We got as a key answer this answer. Breeding is in conflict with several sustainability goals. We knew that, but that's not our base conviction as breeders, but it is their base conviction. And these three arguments are mostly mentioned. There's an extreme focus on productivity. And now all 37 participants in this seminar can say, yeah, but that's not true. I agree. But still, that is the opinion in the outside world. There's an extreme focus on productivity, they say, they think. There are disadvantages for the animals involved. I think we all agree that up to, say, about the year 2000, that was true. And words are used like inbred, overbred, one-sided interest breeding. So the outside world is more negative than we think ourselves. And as I already introduced, the people from the breeding companies said to us, we don't understand those developments. They are too unpredictable. We don't really know the external influencers. And we think it's useful, maybe there's even a need to reach that outside world. Not as a lobby, but for understanding. When we asked, is there a need for a relation between breeders and NGOs to the NGOs? They said, yeah, there is, but it's not the technology which is the problem of animal breeding, but it's the orientation and the consequences of the breeding. I'm not sure this holds for gene editing, genetic modification, and there is some troubles in the technology of reproduction technology in mowed systems, etc. But for the rest, it's rather the outcome than the technology they oppose. And this is an important sentence, the lower red one. This is a citation from the interviews. We asked them, do you trust breeders that animals and other sustainability aspects are in good hands with them? And the answer was, how can we trust them if we don't know them? And that brought us to the concept that there's quite a distance, a gap in knowing each other, in the ethics, and in the information. We will be sharing this more in detail. I'm not giving so many real results now, but I will give 3 lessons and 3 advices. Breeding is seen as part of the problem by outsiders, societal actors in Northwest Europe, especially in the Netherlands, but they are not often seen as a part of the solution. There's hardly any view of NGOs that quite a lot of the sustainability problems can also be overcome with genetic selection. Second, it's a typology. They see us as weighing technocrats. Sorry for offending all of us and you. We are always weighing. We heard a few times. We weigh sustainability aspects against economy, against genetic progress in traits which are of economic importance or important for selling your animals, etc. And we highly focus on the technology of it and not so much on the acceptability or how it is communicated, how it is seen in the outside world. So I don't really see it as an offense, I rather see it as a typology. It's their words, of course. And third, we experienced multiple distances, and this is worked out in the paper we're working on at the moment in more detail. There's a social distance. There's a lot of interaction between NGOs in the Netherlands, Northwest Europe, especially Netherlands, Belgium, between NGOs and producers' organizations, but not with the breeders. So we don't know them, we don't see them. There's not so much social interaction there that people know each other. You're not in each other's networks. Secondly, there's quite a bit of ethical difference, and the weighing technocrats line already said that. We are always talking about balanced breeding. We balance things. And they don't see, they didn't hear so much about, are there limits? Is it necessary to change your animals? Where are your limits? What's your values regarding doing good to animals? And there's technical, I rather should say informational distance. The breeders don't always know what is the societal worries about their activities and outcomes, and the NGOs don't always know what good is done or what activities are performed. And this multiple distance approach complicates any discussion. That's the reason we switched from discussions to dialogues, because discussions are at different wavelengths. That we are talking about balancing and doing good, and they are talking about, but where's your limits? Why are you doing that? Different wavelengths. 3 one-liners advices. 1. The experience of the 3 breeding companies in Wageningen is that we need this type of outsiders, people who are open to talk with us and advise us in a safe environment, to open up our views and to put a wider set of glasses on our noses. For example, one question which was raised in a small audience. An NGO person who would ask you, do you have your key values regarding animals explicit? That's an ethical question with a philosophical background. And the question, in fact, is you are doing things with animals. What's your limits and what's your goods and bads in that? Do you have that explicit? And in several of the projects we are discussing today, ethicists are involved. That's why we involve these people to get acquainted to this kind of questions. Second, it's a bit of an old man's advice. If they, the outside, don't know you, and your business comes into the spotlight, so this doesn't reach us as academics, but especially the industry people. If your business comes into the spotlight, you have the disadvantage of the foreigner, of the stranger. You're an opponent then. You're not a trusted party. You're the foreigner who does something which people don't like. In the swine fever crisis, farmers and veterinarians really touched on this one-liner. Third, that's an advice related to the first one line here. Getting societal partners in your network is a key strategy. And from 2000 onwards, with the projects of Cefabar and Code Efabar and ELSA, etc., we are doing that, but not so intensely, and especially with academics and less with in industry. Getting them into our network is a key strategy. And the term balanced breeding, which we breeders like, and which they hear, is the best team, a very good team, to make a bridge, to see what is balanced breeding then, and why are you doing this, and why are you doing that. But it's also the best team to quarrel, to see where you differ, because we will never be, of course, fully on one line. To finish up in this speed, I have one key reservation. The 3 breeding companies in the bottom of this slide are Dutch-based breeding companies. They're acting worldwide, but the majority of the work was in the Netherlands, a bit in Belgium, a bit in Brussels, with Eurogroup for Animals. So it's quite a bit a Dutch view. And we're not fully convinced whether it fully holds in further Europe or outside Europe, but that's part of the continuing work in the next year, probably. To finish up, breeding in society, I started with the 2 questions, or the statement, be aware of the gap, and should we bridge the gap? Can we bridge the gap? And we explored that from the outside in. We will be detailing on this. This is just a teaser for the World Conference of Genetics next year in America for all of you, or the Ethics Conference in Turkey. Next year we will be presenting both of these if we are allowed there. But the conclusions, yes, there is a gap, clearly. Remember the wavelengths we're discussing on, the ethic differences. And secondly, yes, in dialogue, in interaction, in getting acquainted to each other, it is possible to at least partly bridge the gap. And a background point again, animal breeding is in the midst of society, although they don't see us. We are in the midst of society, and we're dealing with animals, and dealing with animals is very sensitive in my country, is increasingly very sensitive in Europe. It's increasingly sensitive in large parts of the world, especially the Western, but more than that. So beware if you're working with animals. Don't just defend what you're doing, but listen what the opposition is. That's all for now. So I can stop sharing.
Thank you, Karel. It was a very strong finalization to your presentation, but as someone working for the animal breeding sector for such a long time, I totally agree with you that most of the time, I think maybe we're not so good at communicating ourselves in a means of the solutions that we can actually bring with genetics and animal breeding to some of the problems, some of the societal concerns as well. So if we move on to the discussion part now. Can I ask all the speakers to open their videos? And everybody in the audience is also very much welcome to just raise their hands and join the discussion. So maybe with a starting question, I can ask the other three very technical researchers that worked already on the methodologies of new genomic technologies and gene editing, epigenome editing. What are your key takeaways from Karel's presentation?
Maybe I can
Eric?
start with a few
Yes.
words if you want to.
Yes, great.
So it's really fascinating. And of course, there is a gap between society and what we do. But also part of our job is to weight things, as Karel mentioned, and to delve into the details and be very technical and about it. So this gap will probably always be there. And but what's really good and what we need to do better is to listen to society. So be not just defend ourselves, but Karel said that already, but it's what I like a lot. Not just defend what we are doing, but listen to what society is telling us about how they perceive it.
I can
We
also
just
say
lost.
Huh?
Yeah, you go, Matías, but I think we lost Shala.
Yeah. Okay. While we wait for her, I guess the rest can. Yeah. But I have reflection. Also had the chance to hear the work from Karel's group in Breed for Food in the other project that he mentioned, and there we had some discussion later with some colleagues that how sometimes this communication challenge doesn't only happen with we just oh no, okay, there's still 29 participants. I only saw Eric, Karel, and you, and I thought it's just between us now. We sometimes have these challenges with other colleagues from other related fields. So, for example, nutritionists and people in animal production systems sometimes also don't see breeding as one possible help to building a solution, right? And so if we cannot reach to nutritionists, then it's much harder to reach animal welfare NGO activists. So I think there's a lot of people with varying degrees of distance, right? And sometimes a good training to reach someone that it's far away is also reaching people who we are not really communicating well, but they're not so far anyway. So that's one thing.
So Chala is having some connection issues, so I'm just going to quickly take over here. Did anyone answer Armia's answer from the chat? No. He's wondering about if he can get a hint on how to improve two different traits on litter size and growth at once. And then if anyone has any other questions or comments, please feel free to put them in the chat as well.
Okay. That's not directly related to gene editing or epigenetics, right? But it's well, there are tools to do that. So you usually select, for example, a very classical thing, you select on an index that has a combination of both. So you select animals that are good for one or the other, or even better for both. You can check selection index theory for that.
Ebba, is it allowed that I answer to that question too?
Yes.
Yep. Of
Sorry,
course.
I was I'm hopefully back.
Okay. The question which was raised, how to combine those two traits, litter size and birth weight or growth together, that's one of the most prominent examples we got back from talking with people, at least on pigs, because increasing litter size increases mortality too and reduces birth weight, and they're related, of course. And that's a real dilemma. And in the market of selling genetics pigs, That's a difficult one because more fecundous pigs sell better, but have more dying piglets. So how to deal with that? And of course, as Matthias already said, you can optimize between the two. And the real question is, how do you optimize and at what level? At what? How do you weigh out those two? And then it's more logic to take your economy, of course, combined with something you really don't want. And it's not that you do not want large litters, it's that you do not want, if the mothering ability is too limited, or that animals are so sensitive or vulnerable that they die early because they are less than 1 kilo or whatever. So that's the key example of the real dilemma breeding companies have in staying in the market and still doing a very good ethical job. And you see differences between breeding companies in their strategies in that. And it's a sensitive one to discuss in conferences because it's close to market sensitivities too. That's my answer, Ebba.
Thank you. That's great. Anyone else have anything to comment from the speakers or from the audience? I think we got another question as well here. Matthias, yeah, it was you answering in the chat there. But any other comments or questions on that? Or anything else? Unfortunately, I can't really give any good questions because this is way above my head, this technical topic.
Do you hear me? I'm trying to connect, but
Yeah.
Yes.
okay. In fact, I wanted to ask Guillaume, especially about the epigenome editing, because I think there is no actual change in the DNA sequence like the genome editing or some of the technologies. How do you think, I mean, you already gave some ideas about it, but what do you think the trajectory will be for epigenome editing in being uptaken or upscaled with the breeding companies as well? Do you think there could be a market really for that from the farmers and breeding companies?
So the true answer is I have no clue, but I think because it's a new technology and because it's touching sensitive topics such as food safety and so on, people will be very cautious about it. Nonetheless, if the benefit is obvious, I think it could happen. It could be made commercially available and things like that. And it's really my example about messenger RNA vaccination. I was amazed how society accepted this new messenger RNA vaccination and having a foreign messenger RNA injected into our body. And that demonstrates that if the benefit is obvious, then a large part of the society is okay with that. So it really depends. Will there be an application where the benefit is sufficiently obvious to overcome the fact that people will be afraid of having a new technology in their food?
Thank you. Thank you so much. And Eric, how do you think maybe epigenome editing could complement to genome editing, taking into account the work we have carried also in RUMIGEN, because we also have an epichip now from RUMIGEN that is, I think, something very innovative for the sector.
Yeah, but the chip have nothing to do with epigenome editing for the moment. I think that I agree with Guillaume. I think it will be also difficult. For genome editing, I completely agree with Guillaume. We need an example that is that brings many opportunity. For example, I always think that if we had a gene to edit, but Roslin Institute is working on for the avian flu, for example, because every year we are killing many birds, and if we could have some solution even by genome editing for this problematic, but it's the same for the problem that we have had this summer in France for the bovine disease. That was the same. If the sole prophylaxis is to kill all the herd, it's completely terrible. So if we could have other solution, even if they came from genome editing or epigenome editing, I think now at that time the society will move for sure. And that's crazy. I'm completely agree also with Guillaume because actually genome editing is used for medical purpose. So some people are ready to have some of their cells that are genome modified in vitro and re-inject into them because they want to they are real and so they are ready to try many things, even new technology. So this is my
Yeah, I think maybe from the society's perspective, maybe Karel can also comment on it. But when it comes to your health, improving your health and welfare, especially for humans,
yeah,
it
but
is much more easier to
yeah,
accept it. While when it becomes to eating what has been technologically altered,
yeah,
at least that is the perception, is not so. It
but
is a little bit like Frankenstein uh, position. At least this is how the consumers see it because they don't know. When you don't know it, you
yeah,
are
but
afraid of it.
where is the animal welfare when you destroy some herd because one animal in this herd have a disease? That's completely crazy, and this is apparently accepted. I don't understand. Really, I don't
Yes,
understand.
maybe there is room
Yeah,
for
can I
us to
can I respond for
communicate
that
better.
to that?
Yes, yes,
Yeah.
please, Carl.
I don't fully agree with Eric and Guillaume because I think other processes will work in society than the rationale we have in stopping killing animals. This is an improvement. I think roughly there are three processes that will run. One is people primarily look at their own interest. And in the COVID-19 example, it was their own interest at that moment and they didn't understand what they got. Secondly, emotion in animals plays a role. In our parliament in the Netherlands, more than 100 of the 150 seats, delegates, always vote in favor of animal interest things. And that's not unique for the Netherlands only. And the third is, if it's high tech, there's always worries. Worries, do you want to do this to animals? And what's the risks for me, for the animals, for my food, and for nature or ecology, whatever? And then the framing of words which were used by Chela a minute ago of Frankenstein food and so, that frame can kill acceptance. Don't oversee the possibility of acceptance being killed by a simple frame out of emotion or communication. That's all.
Yes, I think really open communication towards the society and societal um, representatives, let's say, is quite important, especially from the researchers as well, because when it depends also on how or who is talking to them. Because I think people still believe more in the researchers and the research behind, especially in European Union, that it is for the common good. And I think it is a uh, bit of push from the researchers to communicate better with the societal forces, societal representatives, I think, but
Yeah,
not
but
limited to
yeah,
that, of
sorry to
course.
interrupt you, but I don't think that we as scientists are the change agents for acceptance. We have to be open, but the choices are to be made by society, by politics, etc. We can give the arguments, but
Yes,
the weighing of the arguments is not up to us.
yes, that's totally true, but I'm talking about like the open communication, like
Yeah,
explaining.
agree.
Yes.
But we are biased.
Um,
Technology.
Yes, of course. That's also another point. Yes. So I think at least in Europe, we have sort of a balance where the consumers are also have the talk. But as we can see, it is still trying to progress from the EFSA scientific opinion given in the last rounds. I think
Yeah.
there is a possibility of progress as well.
Yeah.
I don't know if there are any other comments because I don't see the chat box anymore. If there are any questions from there. If
No.
not, we are then just on time. But Matías, do you have any final words? Because you were more silent, at least when I connected.
Oh, thank you, Chala. I had the chance to talk. Well, just when you were not connected. So
Okay.
thank you.
Okay. Then thank you very much for this nice discussion and also very informative presentations. And a special thanks to Karel for his attendance today and inspiring presentation for us from the Netherlands and the Northwest Europe examples. Things to think about for coming period. So have a great rest of the day and also early a nice weekend. Thank you so much.
Thank you.
Thank you very much.
Bye-bye.
Have a
Bye.
nice day. Bye.